In this video, we will discuss about adverse effect of standard and TCM based CAR-T cells. In the previous video, we have understood about the characterizations of the standard TCM based CAR-T cells and their efficacies. However, the adverse effect was not discussed in the previous figures or in the previous videos. Here, they have explained the adverse effect in terms of the cytokine release syndrome. So first they checked it for the different dose amount. As in the previous video, we have understood that TCM based CAR-T cells amount required is low compared to the standard CAR-T cells for the same amount of efficacies. So when they checked it at a different dose rate, they are able to find out that the TCM based CAR-T cells does not have adverse CRS effect of grade 4. Maximum they can see it for the grade 3. However, for the standard CAR-T cells, they are able to see the adverse effect even for the grade 4. And when they checked this with the same amount of CAR-T cells infused into the patient, the difference still remains the same. That is, the TCM based CAR-T cells can show the adverse effect of grade 3, whereas the standard CAR-T cells shows the adverse effect even of this grade 4. Afterwards, their correlations with this peak of the CAR-T cells are been analyzed, and they are able to find out that for the standard CAR-T cells, when the expansion reached to this peak level, that is correlated with this grade 2 to grade 4 level of CRS effect. Whereas in the case of this TCM based CAR-T cells, even at the peak stage, they are able to see the grade 1 and grade 2 TCM, grade 1 and grade 2 CRS effect in this TCM based CAR-T cells. So this indicates that this TCM based CAR-T cells has lower adverse effect. Afterwards, they check the fever in both the groups, and they are able to find out that both the groups shows fever, but the standard CAR-T cells treated cohort shows fevers at the first stage, whereas this TCM based CAR-T cells fever shows the second stage. And this is also in line with their previous results, where they have seen that TCM based CAR-T cells reach its peak at the second week, whereas the standard CAR-T cells reach its peak at the first week. Afterwards, or carrying forward, they checked the C-reactive protein to check the adverse effect because of this immunological response, inflammations response. To check the adverse effect in terms of the inflammation response, they use this protein, that is C-reactive protein. And although the fever temperature is almost same for both the cases, the CRP proteins is significantly higher for the standard CAR-T cells compared to the TSCM based CAR-T cells, indicating that TSCM based CAR-T cells decouples expansion and toxicity. Afterwards, carrying forward, they wanted to find out what kind of cytokines is playing a role or driving these inflammations in both the groups. So they did the cytokine profiling, and from that they are able to find out that IL-6 is mainly responsible. IL-6 reach its peak stage in these standard CAR-T cells, and whereas in the case of TSCM based CAR-T cells, this is the IFN-gamma that reach its peak. So this indicates that the IL-6 drives the standard CAR-T cells inflammation, and this IFN-gamma drives the TSCM based CAR-T cells. And these things they have further validated or further connected it with the different stages or different grade of the CRS response, and they are able to find out for the standard CAR-T cells, this grade 3 and grade 4 is correlated with high IL-6, and for this TSCM based CAR-T cells, this high IFN-gamma is correlated with grade 1 and grade 2, and also to some extent with this grade 3. So this further indicates that the cytokines this further provides the information about the different cytokines that plays a role in this inflammations. Afterwards, to find out the more detailed insights, So what they did is they stimulate this first. They isolate the CD8 plus CAR-T cells from both the different categories, that is from the standard CAR-T cells and these TSCM based CAR-T cells, and then stimulate them in the CD19 plus stimulate them with CD19 plus targets, and then did the clustering this did this, and thereafter did this single cell. I'm not able to recall that exact name, but the analysis that provide us the 14 different clusters of these CAR-T cells, and out of these 14 different clusters, they are able to see that this IFN gamma is mostly correlated with this panel or cluster 11 and 14 and this cluster 11 and 14 are majorly comprises of these TSCM based CAR-T cells, and whereas this IL-6 mainly comprises of this cluster 1 and cluster 2, and which are predominantly dominated by the standard CAR-T cells. So this further validate their previous finding that IL-6 drives the inflammations in the standard CAR-T cells, and IFN gamma drives the inflammations in the TCM based CAR-T cells. And overall, they wanted to make a point that this TCM based CAR-T cells has less adverse effect compared to the standard CAR-T cells for the similar amount of dose. So this is what they finally conclude from this figure. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.